Today, we'll be looking at how scientists found a new way to control the brain with light. No surgery required. If you're new to this channel, welcome. This is Mr. Singularity, where we explore the scientific and technological breakthroughs shaping the future as we know it. If I had to place the money on the neurotech that earned the Nobel Prize, it's optogenetics. Technology utilizes light from various wavelengths to manipulate the brain. It is a genius mind meld in fundamental neurobiology and engineering that hijacks the process behind how neurons spontaneously activate or are silenced in the brain. Thanks to optogenetics, in only 10 years, we have been able to artificially ingest memories in mice, decode brain impulses that contribute to pain, untangle the neuronal code for addiction, reverse depression, restore primitive vision to blinded mice, and replace horrible memories with pleasant ones. Optogenetics is analogous to a universal brain programming language. Yet there are two serious downfalls: the need for gene therapy and the need for brain operations to insert optical fibers into the brain. The initial mind behind optogenetics is back this week with an upgrade that cuts the thread. Dr. Carl Dieseroth's team at Stanford University, in conjunction with the University of Minnesota, unveiled an improved variant of optogenetics that regulates actions without the need for surgery. Rather, the device shines through the brains of the mice and penetrates deep into the cortex. Through light pulses, the team was able to adjust how probable the mouse was to have seizures, or to reprogram the brain so that it favored the social business. To be clear, we're a long way from science manipulating brains with flashlights. Genetic modification is the secret to optogenetics. Without it, neurons, including yours, don't actually respond to light. However, thinking forward, the thesis is a move in translating a powerful scientific technology into therapeutic treatment that may possibly benefit patients with neurological disorders such as autism or epilepsy. We're quite a long way from that vision, but the analysis shows that it's theoretically within the scope of science fiction. Opto what? We ought to look a bit further into how brains function to grasp optogenetics. Essentially, the neurons work on electricity with an extra piece of chemistry. A brain cell is like a living storage container with doors, called ion channels, that divides its internal atmosphere from the outside. As the neuron gets feedback and the input is powerful enough, the cells unlock their doors. This mechanism produces an electrical current, which then gallops down the output branch of a neuron, a biochemical highway of sorts. At the terminal, electrical data is converted into thousands of chemical ships that float through a neuron distance to deliver the message to their neighbors. This is how the neurons in the network interact, and how the network in turn generates memories, feelings, and habits. Optogenetics is hijacking this process. Using viruses, scientists will connect opsin genes, a specific class of algae proteins, to live neurons. Opsins are specialized doors that unlock under such light wave frequencies which mammalian brain cells can't do. Adding opsins to mouse neurons, or ours, basically gives them the ability to react to light. In classical optogenetics, scientists insert optical fibers close to opsin-dotted neurons to provide light stimulus. Computer-programmed light signals will then invade these freshly light-sensitive neurons in a specific area of the brain and regulate their behavior as string puppets. It's getting hotter. Using genetic engineering, scientists may even fine-tune which neurons have the extra power. For example, either those that encode a recent memory or those involved in depression or epilepsy. This makes it easier to experiment with the synaptic pathways using light, while the majority of the brain is humming along. This selectivity is partly why optogenetics is so strong, although they're not just ponies and rainbows. As you might guess, mice don't especially like being connected to the optical fibers that sprout from their brains. Humans do not, however, have a hiccup in the adoption of the medicinal usage instrument. After its launch, the key aim of next-generation optogenetics has been to sever the thread. Goodbye surgery. The new research team began with the key aim of the Dieseroth team. Let's ditch the need for surgical implants. This is a complicated problem. This ensures that bioengineered neurons within the brain need to provide a responsive and strong opsin door that reacts to light, even as light signals are diffused across the skull and brain tissue. It's like a phone game where one person screams a message from 10 blocks away through numerous walls and street traffic, but you also have to be able to decode it and carry it on. Luckily, the team had a candidate, one so nice that it was a Carmine. Established last year, Carmine stands out in its shockingly quick response times to light and its capacity to produce large electrical current in neurons, about a 100-fold increase from all of its predecessors. Since it is so sensitive, it implies that even a spark of light at its chosen wavelength will allow it to unlock its doors and in turn to regulate neural function. What's more, Carmine shuts down soon after it opens, indicating that it doesn't overstimulate neurons, but still follows their normal pathway of activation. As a first test, the team used viruses to apply Carmine to the region deep within the brain, ventral tegmental area or VTA, which is crucial to how we process reward and addiction, which is also involved in depression. As of today, the only way to access this region in a hospital environment is with an embedded electrode. With Carmine, however, the team noticed that a light source positioned just outside the scalp of the mice was able to accurately cause neural activity in the field. Randomly stimulating light neurons, although amazing, might not be all that useful. The next test is whether it is feasible to monitor the actions of the mouse by utilizing light from beyond the brain. Here, the team applied Carmine to the mouse dopamine neurons, which in this case brings a sensation of satisfaction. Compared to their colleagues, light-enhanced mice were much more likely to push a button to transmit light to their scalps, meaning that the light activates the nerves enough for the mice to sense joy and function for them. In a more complex test, the researchers also used light to monitor a group of brain cells called serotonergic cells at the base of the brain called the brainstem. These cells are known to affect social behavior, that is, how much a human likes social contact. Mice with Carmine-enhanced cells, especially in the brainstem, tend to spend time in their social zone test chamber relative to their siblings who did not have Carmine. In other words, without some open brain surgeries and only a few light rays, the team was able to transform a psychologically ambivalent mouse into a friendly social butterfly. Brain regulation from far. When you think about creepy, you're not alone. The study indicates that through the infusion of a virus that holds the Carmine gene, either through the eye socket or through the veins, it is theoretically possible to regulate something as integral to personality as sociability through nothing but light. To emphasize my point, for now, this is only feasible in mice. Our brains are far bigger, which ensures that light reflecting across the skull and reaching far enough becomes much more difficult. And again, our brain cells typically don't respond to light. You'd have to volunteer with what amounts to gene therapy, which comes with a variety of potential problems, before it might actually function. So hold the tinfoil hats off. Scientists can't turn an introvert, like me, into the laser extrovert yet. Yet it's a great jump into the future to unravel the inner workings of the brain. Thus far, attempts to sever the optical cord for optogenetics have culminated in a kneecapped capacity to go deep into the brain, restricting control to only surface brain regions such as the cortex. Other approaches overheat delicate brain tissue and result in injury. Yet others operate as DOS systems in the 90s, with a considerable pause between the order, activate, and the neuron reaction. While not quite perfect, this brain control OS solves these problems. Unlike Neuralink and other neural devices, the research indicates that it is possible to regulate the brain without surgery or implants. The only thing you need is light. If you made it this far in the video, thank you, and welcome to the end of the video club. What's your take on this? Let me know down in the comments below, and check out one of these other videos. This has been Mr. Singularity, and I'll see you on the next one.